Thank you very much, Svetlana. We at LODAT really appreciate the opportunity of partnering with CPHI and sharing our learnings and our scientific research and our skills with the folks here and the attendees at CPHI. If any of you were in the morning session, the market opportunities session, we went through a very broad range of specific market opportunities and an overview of what natural extracts are and specific case studies. This particular session is on quality assurance and technical standards. And I'm going to go through a very brief overview, and then I'm going to introduce a very well-versed and extremely knowledgeable scientist, Dr. Ranjan Mogre, who's part of our team, our Chief Science Officer, and he's going to go through a lot of the specifics and the technical details on quality assurance. So LODAT is our company. It stands for Live One Day at a Time. We are a science-based firm, and we like to say we're very innovative. We integrate 21st century technologies with an integrative approach for reasonable cost. So what we basically do is all forms of generic pharmaceuticals, nutraceuticals, which is natural extracts, dossier and tech transfers, technology or turnkey solutions for specific sites, as well as API ingredients. If you want to know more about our firm, we're over here at the end of the presentations as well. We'd be more than happy to speak to you as well. So our competitive advantages, just very briefly, is that we are a US-based firm. And this is our business model, that we have a very flexible manufacturing as well as multiple pricing structure. We do have our own proprietary manufacturing process, which is what really this particular session is about, and how we develop that process, as well as proprietary blends. Everything from halal compliance to a very high efficacy and stability, and research and backup studies on all of our claims. And one of the reasons we're here, obviously, is to share our information, but we're also looking and always in search for good partnerships and business development partnerships. And we're committed to working in different markets and different regions. And again, we're here to hopefully develop those partnerships. So before I introduce Dr. Mogre, I wanted to share with you really what the gist of our new technology is, if you will. And in this instance, the product is the process. What we've done at LODAT is we have created a matrix, if you will, of 3 primary types of processes. The first is the nutraceutical process, which is consumer-based. Placebos are common. We have the SCFE in place, and we're going to really go into that. Dr. Mogre is really going to go into that. Pharmaceutical processes, most of you understand those processes, so I'm not going to go into too much of that. And then also alternative processes, which is what we talked about in the morning session. So that intersection is what's called thermogenesis, or AY thermogenesis. And it's by combining these methodologies that we've developed a very objective, evidence-based procedure to market ingredients and to market natural extracts. So with that, I am going to introduce Dr. Mogre. He's going to talk about supercritical fluid extraction, or SCFE, and specifically those processes in that, as well as some of the data to support both thermogenesis and the latest technologies. So, Dr. Mogre.
As Rajiv mentioned, we have been really talking too much about, synthetic products these days and the side effects of the synthetic products. So probably we would be talking now and more in future, more of the natural products and the different ways we can get the natural extracts and try to use these natural extracts as an alternative medicines, or even as the main API, which we are commonly used to as the generic APIs or synthetic APIs. I would not really go in too much of technical details because I understand, the audience could be from varied background, talking about the technology in general and the application of the technology, the application of the extracts, how the formulation is developed and how the analysis is done in terms of how the quality is assured, and then different aspects of marketing, which Rajiv already spoke in the morning. So in brief, I'm going to talk about, what are the natural herbs, spices, what the market potentials of it, the different methods of extraction, why particularly we need extracts to work with, what are the applications and what are the different conventional techniques which is used for extraction, and little bit in details about the supercritical fluid extraction technology, which is probably the technology now getting much specific credit for the new technique that's used for it and the applications of this extract. In terms of the quality assurance, I'm sure most of you would be familiar with the different techniques which are used, like the HPLCs or the GCs, and there are faster techniques like the RSLCs or UPLCs. I would only briefly touch upon this. Briefly touch upon the various aspects of formulation development and how the product is actually formed and goes to the market. In short, if we just recap a statement which was almost, I would say 2,500 years ago, which was made by the Greek philosopher Hippocrates, said that let food be your medicine and medicine be your food. Which itself reflects the changing mind of the people now, which it's taken almost 2,500 years for us. We started talking that we take the natural food as the preventive medicine or something. So that really goes beyond the proof that the philosophers are more often than not they're correct. This is a picture which depicts how actually the whole world is completing a cycle. If you look at the cycle of clothing, the earlier days, in the Stone Age, people were hardly using any clothes. They were sometimes using natural leaves. Then they started growing cotton and started wearing cotton dress materials, then into the semi-synthetic ones. Over a period of time, we have gone beyond 2,000 years and we are into the designer clothes. The designer clothes are actually very soon, there's a fear that we can go down to the natural leaves here, because the designer clothes, you hardly have, a long piece of cloth on you. You're probably going back to the Stone Age again, which is a quite resemblance to the foods, to the medicines what we have. We used to have the natural foods, the leaves, the fruits, then we started cooking, then we started semi-processing. Now we are back to natural extracts, which we are going back to the natural fruits. So these two cycles of clothing and the foods are a complete picture of how we are changing the mindset from the original Stone Age to the original natural foods, going back all over the complete cycle to the natural things, changing in the meanwhile the different aspects of life. Now, if you look at the plant kingdom, which is very broadly, if I can classify into the herbs and spices, most of us are very familiar with the spices. There are different spices like the cloves, the cinnamon, the black pepper, everything you name it. They have a different and very specific application in day-to-day activity. They could be used not only as for the culinary purposes in the food, but most of the spices can also be used as the homemade medicines for small things. Like if you just have a cough or cold, you can have the spice extract and you can get rid of the cold and cough symptoms. But the major part of the plant kingdoms are the herbs. There are different herbs which we can start learning more and more about the herbs and their applications as medicines, the applications of cosmetics, and then the applications of dietary supplements or nutraceuticals, or you can call them as alternative medicines. Then there are foods, and the aromatherapy is again a growing area of the plant kingdom. The point I would like to mention here about the reasons of extraction is because as the growing requirement and the consciousness of scientific guidelines, you have to have the specific quality of everything. If you only stick to the spices or herbs, the quality of the herbs can change with time and different locations, which is not acceptable to mankind now, because the level of guidelines and level of consciousness we have developed over a period of past so many years, which indicates that we need a specific quality of the material, and that can be controlled not by the common natural product but by the extracts what one can use. If I had to store the natural raw material, like if I had to store the herbs or the spices or the plant material, I need a huge storage area. And if I had to control the conditions of the storage, I would need a huge energy input into that. Instead of that, if I start working on the extract of the natural product, I would need a small area. I can control it much better in terms of the quality parameters, which I can guideline and check it time and again. The other thing is there would be drawbacks, when you pick up the natural material from January to March harvest and October to December harvest. There would be differences in the natural product composition, and you would be in practice you would be using a natural product which is actually harvested in January to March, which can carry different properties. So in order to get over all these drawbacks, it's very important we start working on standardized extracts. Now, once we say that, okay, we need to work with standardized extract, we need to know, what are the different options available to me for doing the extraction of the components which I need, which I know that, the component carries the medicinal value, the component carries the cosmetic value, or the aromatherapy applications. So how do one do that? There are well-known methods which are centuries-old method, like the aqueous extraction or infusion, which are called as the water extracts. The limitations of water extracts is, whether you say a natural product or you say a synthetic product, in true scientific terms, they are chemicals. So these chemicals, depending on the nature of the chemical, depending on the structure of the chemical, will have the solubility properties. So if the product is not soluble in water, you would not be able to get in the aqueous extract. This will be a defunct method if you want to extract something which is not soluble in the same. Then the next method one can use is the solvent extraction. The issue that comes up with the solvent extraction these days is, the guidelines from the regulatory authorities say that, I cannot have a product which contains more than specified limits of organic solvents. So although this is a very useful method, so you can vary the solvents from very non-polar hexane to polar methanol or ethanol, the guidelines will tell you that, the limits of the solvents which are left behind in the extract should be very minimum. Otherwise, it would be injurious to your health. Then there is a steam or hydrodistillation method which was used and developed, but the application of this technique is mainly for the essential oils. It's not for the extracts. So these products which are obtained by steam or hydrodistillation can be used in the aromatherapy, can be used in the perfumery industries, but not in the medicinal product. So what is left now is try and work on method which would be satisfactory to both in terms of the applications and in terms of the regulatory requirements. So there the supercritical fluid extraction technique that comes into picture. Now, what is this technique? It's a simple technique, and why it has come out? The reason it has come out is because there are increased business opportunities. If you see now, probably 10, 12 years ago at the CPHI itself, there would not have been too many stalls of natural extract suppliers. Over a period of last 10, 12, 15 years, the number of the natural extract suppliers have suddenly started growing. So there are more business opportunities for the natural extract suppliers. The guidelines, which I just mentioned in terms of the solvent residues, in terms of the quality of the extract, are making the technique that one want to use it in the extraction more scientifically correct. the better specifications. You talk about the generic pharmaceuticals or the small molecules people talk about. The simplest medicine one can talk about, paracetamol. It has a very well-defined specifications. So the mindset now is whether you have a synthetic small molecule or the newest molecule or even the natural extract, the specifications are going to be very stringent. And if I decide to have a very stringent specifications, I should work with a technology which gives a very high quality of the extract. The best quality ingredients, depending, I'll come down to my presentation, you will realize what it meant by best quality ingredient. There would be all these methods which can be used to manufacture the same component, but if you extract that with supercritical, the quality comes out to be the best. Now what is this technique? It's basically, it takes care of the physiochemical properties of the solvents. We all know every compound in the nature is classified into a solid, liquid or gaseous stage. So if you take a gas and try and compress it, that means try and apply pressure on it, the gas can be liquefied. If you liquefy a gas and start heating it, the liquid starts going back into the vapor stage. But in the vapor temperature pressure diagram, if you look at a point which is called as a critical point of a component, now this point actually determines what will be the nature or the property of the component. At the critical point, no matter however higher pressure you apply, the substance will not get liquefied, which behaves different than a normal physiological properties. So it remains in the gaseous stage. So this temperature at which whatever higher pressure you apply, it will still remain in the gaseous phase, is temperature called as the critical temperature of that compound. And the pressure is called as the critical pressure of that compound. So what anybody in terms of the extraction techniques try and use it is this area in your pressure temperature curve. This area indicates that the substance, what you want to use it, will always remain in the gaseous form. Actually, to get into more details, it's not the gaseous form, it is called a dense gas form. The dense gas form indicates that the substance is neither gaseous nor in the liquid form. By being in this state, the property of liquid as well as gas is utilized for the extraction. We all know that the gases can diffuse very easily compared to the liquids, but cannot dissolve any component like liquid. So you need to utilize the solubilizing property of the liquid at the same time using the diffusion property of the gas. And that's what actually happens in this area. It remains into a dense gas stage. Now there are certain parameters of different compounds which could be used for supercritical fluid extraction. If you look at the entire list of solvents from methane down to water, this water is the most simple solvent one can think about it. But if I want to use water in supercritical conditions, I have to heat it to more than 400 degrees centigrades. Okay, so that creates a problem of my energy which I need to provide. At the same time, if I look at carbon dioxide, number one is it's a physiological gas. So we all know, the body knows carbon dioxide, so it's not a poison to the body, it's not an unknown compound to the body. But most importantly, you can convert carbon dioxide to supercritical stage at 31 degrees Celsius, which is almost the room temperature. So at 31 degrees and about 74 bar, you can convert gaseous carbon dioxide into a supercritical carbon dioxide. The other problem with the water is the most hazardous solvent at the supercritical stage. It becomes highly acidic and start corroding even the most brilliant stainless steel. So water cannot be used. Otherwise, we all know that water is a nice solvent. It's a chemical which is harmless to the body, but at the supercritical it behaves differently. So if you look at these parameters, you would find in the literature 99% of the application using supercritical fluid extraction is only based on carbon dioxide. We all drink coffee and we all drink decaffeinated coffee. I'm sure most of you would be doing that, but few of you would be knowing that the decaffeination of the coffee is 100% done only by supercritical fluid extraction. So that's the application which started almost 35 years ago, and now you have the supercritical fluid extraction being used for all natural extracts. Why supercritical fluid extraction? It's a GRAS process. GRAS is generally regarded as safe because there would be some hazardness of the process which are still unknown, but there's a possibility somebody may come up with something. So it's always called as generally regarded as safe. It's a specific term used by the regulators. Tightening of government regulations on solvent residues. You know, you talk about methylene chloride, you talk about isopropanol. You know, you need to have the solvent residues below ppm level. So if you do not have the solvent level below this, the regulators will say you cannot use the compound in your drug product formulation. The consumer concerns over chemical solvents. You know, we all go to the market and say, oh no, this contains a chemical. So these are the concerns of the consumer. So that's again a cause of you know, using supercritical fluid extraction. Increased demand of higher quality products. We all want to know that, you know, we don't want chemicals, we don't want pesticides, we want pure material. So that's all these things. An increased cost of energy. If you're going to use solvent, you would have to heat it, so there will be cost of energy, and that would start increasing the cost of the material. Now, how is the whole process really works? It's pretty simple. It's nothing different than any organic or any water extraction technique, but the only thing is it's done at much higher pressure. You know, you have the extractor, which is a stainless steel vessel. Now, this vessel is designed to withstand a pressure up to 700 bars. So it's simple. It's about a couple of inches thick wall, and there is no welding on it. So it's meant from a seamless tube. You have the pump, which is actually the high pressure pump, a storage tank which says that it stores the solvent one we want to use, in this case carbon dioxide, and there are different separators. You know, this separators indicates where you are separating the product of interest. So you take your raw material which you want to use it for the extraction, you pump carbon dioxide, you have the carbon dioxide at temperature beyond 31 degrees, pressure beyond 74 bars, and you start pumping in, it will start extracting the product. The only thing here is different than any organic solvent extraction or water extraction is that based on absorption. This is based on adsorption. So the solvent, in this case the dense gas, actually the material of interest get adsorbed on the gas molecules, and that is carried. At the separation level, you reduce the pressure. What it does, it precipitates your component of interest in separator 1. You can change the parameters and get additional component in separator 2. The carbon dioxide gas can be recycled back. So the whole solvent which you use it, this carbon dioxide here, is recycled. So that adds to the benefit of the process. So you don't actually leave a lot of carbon dioxide to the atmosphere. Now, how do you compare this, the conventional extracts against the supercritical extracts? It's simple. You have the solvent contamination. Here you have 100% solvent-free component. Second thing is near natural. What it means by using the term near natural is, for example, a very simple example I often quote is you take a clove. A lot of people are used, or even cardamom. Most simple spice every one of you probably would be using. If you use the smell of that clove or cardamom naturally, and if you take the smell of supercritical carbon dioxide extracted cardamom or clove, it will exactly resemble like the natural material. This may be deformed because you involve the heat, so your natural product can get deformed in conventional method. They are so-called as cheaper products, whereas supercritical products are called as expensive products, which is a myth. Because if I consider the batch economy of my end product, because of the high quality and high purity of supercritical fluid extract, I need small quantities of that. So overall, on absolute terms, the supercritical extracts become cheaper than the conventional one. The highest quality against the quantities. So, this is a pretty standard or general observation is they are required in terms of about 30 to 50% of natural or conventional extracts, simply because of the highest quality of the extract. Where are the areas? Which are the areas the CO2 extracts could be used? They could be used in medicines, cosmetics, dietary supplements, flavors, fragrances, and aromatherapy. These are some of the commercially used CO2 extracts. The cinnamon bark oil. Each one of us probably go down and have the bread or the bakery product called cinnamon roll, the most widely used bakery product around. Then you have the ginger or the fenugreek for the bitterness in terms of diabetic formulations. Then you have the sandalwood oil in terms of the cosmetic applications. And now in terms of the dietary supplements or nutraceuticals, you have the curcumin, you have neem, you have phoscolin, and you have pinacic acid component called pomegranate seed extract. Now, you have done the extraction. How do you actually qualify it? How do you actually say that the product is good in terms of the quality? There are different techniques. We all know the HPTLC is one of the oldest techniques. Then you have the gas chromatography, mass spectrophotometry. You have the HPLC MS techniques which you use for it. Now, if you consider all this, it gives you most extracted natural compound analysis. But there are limitations in terms of how one can certify these products. The limitations are because the availability of the standards. For the small molecules or the generic molecules or the regular pharmaceuticals, which is in practice for a number of years, over the period of last 25, 30 years, there have been various companies producing standards to quantify and assure that, my drug is this pure and my impurities are this and all these things. But these are the limitations in the natural products. These are not limitations of supercritical extraction. Any natural products, the availability of the standards actually starts creating hurdles and the obstacles in quantifying different components in it. The monograph methods, I'm sure you all understand. There are USP, there are BP, and there are JP. With these pharmacopoeial monographs, there are hardly any documentations about these natural extracts in this monograph. It's happening now, but not to the greater extent. These are some of the examples I would like to share with you. Lutein, I was mentioning about the cosmetic products. Now, lutein is an extract from marigold leaves. The application of lutein has been increasing for last 5 years. Initially started as an antioxidant product. Now it is used as a nutraceutical product for AMD applications. The age-related macular degeneration of your retinoid membranes. Lycopene again is an antioxidant from tomato skin, which both of these products can be used as an antioxidant to prevent the carcinogenesis in the body. It's as a natural extract applications. Now here, the HPLC analysis of these two components indicates they're absolutely pure in terms of the quality. Now that's where the limitation starts, and the expertise in the analytical chemistry or the analytical technique that needs to be done has to be widely appreciated. That's the challenge of the chemist now. The example here is of piperine, which is actually the black pepper extract. Now, the piperine is, we all know, we all use black pepper in our day-to-day food products, but the medicinal application of piperine is taking larger steps now. It's widely studied, known bioavailability enhancer of other compounds which one likes to use it as the product, as the medicinal product. Phoscolene, which is again extract from roots of a variety, which was probably patented almost 35 years ago by normal method of extraction by Hex, but now it's available for supercritical fluid extraction. It's a product which is used in the formulation for anti-obesity products development. Again, if you look at the analysis which my colleague sitting with us is has done it almost 10 years ago. Yeah. So which says that the product looks quite clean in terms of the purity of it and the reproducibility of the quality of the product. Curcuminoids, we all have been talking a lot about the applications of curcumin in dietary supplements or in the medicinal applications or in the cosmetic applications. We all know that. It's not actually the curcumin as a single component in any of these compositions. It's a mixture of three components. The most potent in the three being the curcumin, then there is dimethoxy, and then the bis-dimethoxy curcumin. What one should note, that why I've been emphasizing on supercritical fluid extraction, is in the natural composition of curcuminoids in the turmeric rhizome, there's a specific percentage of these three components. If you use any of the common technique like the solvent extractions, which we all know, you all extract curcuminoids, but the ratio of these three components varies, and it is important to have those three components in the same ratio to have the most significant effect of the formulation one develops with. Between these two slides, I would like to point out a major difference what the technology has done over a period of last 5 years. If you look at this slide, which says that the time taken to analyze it and extract is almost 26 minutes. The technological developments in terms of the HPLC over a period of last few years is done that, replacing the normal HPLC by RSLC or UPLC, you can analyze the same formulation, same components in about 5 minutes. As a common person, you would say, what big deal, you change from 26 minutes to 4.5 minutes. It's only, you save 20 minutes. But when you are into production, you are into formulation, manufacturing of this product, you need to find every minute in your day-to-day requirement to make sure that the quality of the formulation you do is completely perfect. So there would be more batches coming to, there will be more samples coming to, and you have to minimize the time of analysis, and that's what actually these two slides explain. This is a lot of us are aware that as a natural sugar substitute, we all talk about stevia extract. This is the HPLC of the active component in stevia extract called steviosides, and the other components which are very clearly identified in the analysis. We all know ginger in terms of the medicinal application of ginger, and we talk about the extract in terms of formulation, the ginger ales or formulation of ginger lozenges. This is a very I would say interesting slide of ginger analysis. The ginger in terms of the components in the ginger rhizomes, there are two separate components in it. One is the ginger oil, which is very nice smelling component but doesn't have the pungency of ginger, so it has a different applications. And there are ginger oleoresins, which are actually the pungent component of the ginger, and that could have a different applications. The ginger ale actually does not have the oleoresin in it, otherwise we all cannot drink ginger ale. It becomes a very spicy component. So you don't add ginger in the ginger ale, you add ginger oil in ginger ale. That's formulated by using emulsifiers in water. You have done a good technique of extraction, you've done the analysis and found that the components are good, you can formulate it, and there are different ways of doing the formulations, which is the composition which contains the API in terms of natural products and natural extract, and you need to make it as simple product so that you can have the correct delivery to the people. The goal and objective of the industry, the regulators and the practitioners, the regulators also should have the same goal, that the development has to be done to create substantially and support substantially higher number of patients to make it more safe and effective medication. Before we talk about formulation of a product, whether you work with natural extract or you work with the synthetic drug, you still have to consider these points, which says that what are the regulatory requirements, what are the R&D hurdles, what are the potential markets. Because if you do everything and there is no market, you would only end up losing money, and then you would start worrying from where do I get the finance. So these are the 4 parameters you should first consider before you start doing any formulation development activity, whether it's a generic molecule or the supercritical extract. Whatever you did so far is probably the simplest aspect of a drug. You would realize it by looking at the number of steps that are involved in the formulation development. And each step actually takes weeks and months to complete. The simplest statement I can make at this point of time is from the paper to the market. If I have to bring a drug, it would take nothing less than 5 to 10 years. Why 5 to 10 years? You would realize it by going through all these things. You have to go through the literature of the components, what are the patents on that, then whether the materials are available, what are the design of experiment one need to do it. Then there will be huge amount of work one needs to do before you end up doing any formulation. And these steps do not alter for any API. These steps remains the same, whether it's the natural extracts or synthetic pharmaceutical. I guess once you have developed the product, you go through the stability of the product, which is common like any any generic components, and then you start selling the product. I think that's all I can share with you, and if you have any questions, I would be happy to answer them. Thank you.
My name is Imo Fiebrig from Explicat. My question is, if you use CO2 for extraction, would you be extracting mainly the lipophilic components and not the hydrophilic ones, or how would it be?
I'm pretty sure you're used to supercritical extraction technique. The normal way, the solvent solubilization category that works, CO2 being a very non-polar solvent, you would imagine only the lipophilic components to get extracted. But if you can increase the pressure, make it at higher pressure, there's a possibility that one component which is dissolved in carbon dioxide, the whole combination start working as a co-solubilizing phenomena, and you can start extracting the higher polarity components in it. The second way is you can always add other organic solvent, like you can add water to it, you can add ethanol to it, to a percentage from 0 to about 10%, which starts making the solubilization of higher polar components much easier compared to using simple single carbon dioxide. Because that's one of the techniques used in caffeine decaffeination. You first remove the flavor of coffee by using carbon dioxide, and then pump in CO2 and water to remove caffeine from it. You have actually the residue which is left, which doesn't smell like coffee, which doesn't taste like caffeine. What you do is whatever smell you have removed by carbon dioxide, you reload that on the residual coffee, and what we drink is actually decaffeinated coffee is that, and the caffeine goes into the colas. Thank you so much.